So continuing on with chapter 15, how do we get an orientation where the N terminus is cytosolic and the C terminus is in the lumen? Well, instead of having a signal sequence at the beginning, you just have that internal start sequence, what was the stop sequence before, but really it's just a run of hydrophobic amino acids. So these hydrophobic amino acids will still get recognized by a signal recognition particle and draw you down to the translocation channel. And since you've already made a whole bunch of N terminus, that's going to still be stuck outside. Now, the signal sequence, the internal signal sequence, is going to act as the transmembrane portion of the protein. And you'll continue to feed the rest of the protein into the lumen and thereby flip your orientation. And then besides single pass transmembrane proteins, first the other one was type 1 and this one is the type 2 transmembrane protein. Besides those single pass transmembrane protein, there are also multi-pass transmembrane proteins. So how do we do that? It's a series of start and then stop and then another start and then another stop, and you end up with 2, 3, 4. The most common gene in your genome is the largest family of genes in your genome is 7-pass transmembrane proteins, what we call the G protein coupled receptors. So you just have to have a start, a stop, a start, a stop, a start, a stop, and a start. And then you'd end up with a 7-pass transmembrane protein. And whether that is type 1 or type 2 would depend on whether you had that N-terminal signal sequence or not. So you could have a multi-pass transmembrane protein that is type 1 or type 2. So watch this video, and it's going to talk about like multi-pass transmembrane proteins and how we just basically stitch them into the membrane of the endoplasmic reticulum. In addition to the proteins that go into the endomembrane system, we've also got import into other compartments. Like, for instance, the nucleus. The nucleus has lots of proteins in it. All of the ribosomal proteins have to get enter into the nucleus to go to the nucleolus to form the ribosome with all of the ribosomal RNAs that are already in there. We've got transcription factors. We've got the machinery for transcription. We've got the machinery for DNA replication. We've got histones. There's a whole bunch of proteins that have to get into the nucleus. But let's think about this. Translation happens in the cytosol. So these proteins get made in the cytosol and have to enter into the nucleus. And so, so how does that happen? It goes, the proteins go in through these nuclear pores. And so the nuclear pores, so just to remind you, the nucleus has a membrane, and it's actually a double membrane. So it's 2 membranes thick. So you've got phospholipid, lipid, phospho, an intermembrane space, and then phospholipid, lipid, phospho. And there are nuclear pores that span across both of these membranes and allow for entry into the nucleus. And it is highly regulated. We can see here on the electron micrograph that the nuclear pore has a whole bunch of proteins right there at the annulus, that very dark spot, and then the darkness extending out from either side of that very dark spot. That's all proteins, because it's obviously electron dense, and so we know that must be proteins. And so we've worked out the basic shape of the nuclear pore complex, and it turns out that there's like a basket on one side and fibrils on the other side. And so you can look at these nuclear pores on the scanning electron micrograph, and we can see these fuzziness across the nuclear pore, and that fuzziness is these fibrils that are extending out into the cytosol, and they are gatekeepers. They decide whether something enters or not, and the only way that something can enter into the nucleus is if it is attached to a transporter protein. So there's a nuclear transporter, and it recognizes some sequence on the protein that it is transporting, the cargo. And this whole process is highly regulated. So when there's a cargo that needs to be imported into the nucleus, it binds to the receptor protein, and so this transporter moves it across the nuclear pore complex, and then it needs we need to get that transporter protein back out to the cytosol, or else it can't do the next round of job. So the way that the cargo comes off and the protein gets moved back out to the cytosol is that it binds to another protein called RAN. And RAN only binds to the transporter protein in the presence of GTP. So when RAN GTP binds to the transporter protein, it displaces the cargo protein. The cargo protein comes off, and the RAN GTP bound transporter protein then exits the nucleus. Now, the GTP gets hydrolyzed. The RAN just does this on its own. It doesn't need another protein to cause the hydrolysis. But when GTP goes to GDP through GTP hydrolysis, now the GDP bound RAN does not like the transporter protein and dissociates from it. And so this RAN GTP, RAN GDP cycle is how we move proteins from the cytosol to the lumen of the nucleus, and I would really like for you to learn this RAN cycle. So that's something that I usually test on. Mitochondrial transport is a little bit different than into the ER, but it's not exactly the same, but it is gonna rhyme quite a bit. So watch this video. But what is the video going to show you? Basically, it's going to show you that how proteins enter into the mitochondria. Remember, they have a different signal sequence. So there's a signal sequence that says go to the ER. There's a different signal sequence that says go to the mitochondria. And the translocation channel on the outer membrane is very, very similar to the ch15 protein. The receptor is very, very similar. The exact sequences are different, but the mechanism is very similar, and that just makes sense because of where mitochondria come from. These are membranes that are descended from the phagocytic vesicle of the original symbiotic event. And so this transporter protein is very similar in its action. One thing that we want to note is that the entire protein got made while in the cytosol. So it was globular, and then the signal sequence was recognized and went to the receptor protein on the mitochondrial membrane. So it had to get linearized again. It gets straightened out like it was when it was being made. Then after it's linearized, the translocation channel, the receptor, and the cargo are going to diffuse laterally until they find another translocation channel on the inner membrane. And once the outer and inner membrane channels find each other, then the protein can be moved into the mitochondria. It refolds back into its globular conformation, and then the signal sequence is cleaved. So if you learn import into the ER, then all you have to do to learn the import into the mitochondria is just learn the differences. So learn the ER first, and then when you get to the mitochondria, you can do it by analogy and just learn the differences between mitochondrial import and ER import. In class, I talked a little bit about the pulse chase. And so just to remind you that in the old days, we used a lot of radioactivity. And so we could tag proteins that were being made in the ER by adding radioactive sulfur, because every protein is going to have a sulfur in it. And so we can label proteins by giving a pulse of this radioactive sulfur, and then we can watch how over time those proteins move from the ER to the Golgi and then to vesicles, which ultimately bind to the cell membrane and are secreted from the cell. This is what we call constitutive exocytosis, or the default pathway is to secrete from the cell. These days we don't use a lot of radioactivity, because now we have a different kind of tag, and that is the green fluorescent protein. Green fluorescent protein came from a jellyfish, and we can use the sequence to make that protein, put it onto the sequence for the protein that we're trying to track, and now the protein in its native form has this fluorescent tag on it. So we can see these HeLa cells under light, and then under UV, and under UV the green fluorescent protein starts to glow. The excitation originally was UV. Now there's lots of different excitation wavelengths and different colors. It's not just green anymore. There's a whole bunch of different colors. But like I said, it came from a jellyfish in the northern Pacific Ocean around Washington and Oregon, and Shimamura isolated it. Marty Chalfie was the first person to tag a protein and put it into another live animal. That was C. elegans. And then Roger Chen has now created lots and lots of different colors. So we can attach these green fluorescent proteins to the protein that we're trying to track, and follow them through the pathway. So move on to video 3.